Okay, so this is the longest section about muscles. And this is where we're going to learn how muscles work, how they actually contract. This is one of my students' favorite things to learn about, and they really understand it well. So hopefully, and they just like it. They think it's interesting. So hopefully it is the same for you. Okay, so we're going to learn how muscles work, and this will help us understand what happens when they're strained. Okay, so muscles, depending on the person, are nearly half of our mass, and they use a ton of ATP because they are converting energy. So we're going to look at types of muscles, characteristics of muscles, and how they work, how they contract. Okay, anything with the terms myo, mice, or sarco has to do with muscles. So if you see those prefixes, that's muscles. An example is sarcoplasm is the cytoplasm in muscle cells. And muscle cells are really weird shaped. They're different from other types of muscles or types of cells that we've seen. There's 3 types of muscle tissue: skeletal, cardiac, and smooth. The skeletal and smooth are longer, and they're considered muscle fibers, and the cardiac muscles are not. And we did look at these in the tissue chapter. So skeletal muscles are usually the ones that we learn most about how they work and what they look like. They are, the tissue is packaged in skeletal muscles. A muscle is considered an organ because it's made of lots of tissue, and muscles are attached to bones and skin. They are the longest of the 3 muscle types, and they have striations. They are considered striated, which we've seen. They are also considered voluntary because you have to think to contract them. And they contract quickly. They tire easily, unlike the other smooth and cardiac, and they are powerful. So if you see skeletal, striated, and voluntary, those are kind of synonyms for skeletal. Cardiac muscles are only in the heart, and you do have other types of muscle in the heart, not just cardiac, but that's your heart walls. They are also striated, but they are involuntary, so we don't have to think about our heart beating. It just beats automatically. And they they interact differently with the nervous system, a little bit differently than skeletal do. And you have cardiac, striated, and involuntary as synonyms. So cardiac muscles are also striated like skeletal. Then you have smooth muscles, and they are not striated, hence the name smooth. These are found in your organs. These are involuntary. You don't control them. They help move stuff around your body. So here are the 3 types. You can see that skeletal and cardiac are striated. Those are the little striped appearances. And then smooth muscles are not striated. Skeletal muscles are long cylinders. So they are like, one muscle is like a long cylinder. They're really weird. They have lots of mitochondria, and they also have lots of nuclei. And then cardiac muscles aren't as organized looking. Like the strips the fibers are kind of more disorganized and branching. And then they typically have 1 or 2 nuclei. And then smooth muscle only has 1 nucleus, and they're kind of spindle shaped. They're not like striped at all. So my picture's covering it, but you can kind of see the spindle shape. Okay, so there's 4 characteristics that all muscles share. They have excitability, so they respond to stimuli. They contract, which means they shorten. They can be stretched, and they are elastic, so they can recoil. Okay, so 4 important functions that muscles do. They produce movement, so they move our body. They move stuff around our body, like food and blood and poop and pee and all of that, babies. They help us maintain posture and position, stabilize our joints, and then they generate heat. So most of the heat that our body generates is from our muscles. Okay, so there's different parts to a muscle that we're going to look at, and we'll mostly look at skeletal muscles. They have nerve and blood supply, they have connective tissue sheaths, and they have attachments. So every muscle has a nerve, artery, and vein. That's no matter what, because they need a nerve to be able to contract, and then they need an artery to provide them with nutrients and oxygen, and then a vein to remove CO2 and waste. And muscles take a ton of energy and oxygen. Like a lot of the energy that we consume goes to our muscles. Okay, so each muscle fiber has some different sheaths that like connect it or contain it. One is called epimysium, and that surrounds the whole muscle, and it blends with the fascia. And then perimysium is like surrounding the muscle tissue, and then endomysium is in the inside around each muscle fiber. So it's like 3 layers. You can kind of think of it like a Twizzler pull and peel, like you have the whole thing and then you start peeling it apart, and each of those has a little connective tissue that covers it. So here is your diagram of that. So the epimysium is on the outside. Epi means above, and then peri means like within. So that's if you zoom into one of these fascicles. And then one muscle fiber has endomysium like around it, and to me it looks like a pack of cigarettes, like a whole bunch of little cigarettes. And then these are the cells, and they're made of protein primarily. Your muscles are mostly made out of protein. So it just keeps getting smaller and smaller and smaller until you get to a muscle fiber, and they're each wrapped with these different structures. Okay, we already talked about origin and insertion in chapter 8. So muscles are attached to the insertion, and then the origin is like what they're anchored to. And the attachments can be direct, which are fleshy, or indirect, which is like a tendon or something like that. Okay, so here are like the 3 levels zooming in. This is like your big muscle, like this is your bicep. And then if you zoom in, this is one fascicle and it's wrapped in perimysium. So there's epimysium around the whole muscle. And then you zoom in and this is a fascicle and there's perimysium. And then if you zoom in even further, this is a muscle fiber. And you have many of these fibers. And that's where the contraction's actually happening. So this is your endomysium, and then inside of that you have sarcolemma, and right here's a nucleus. And so the thing is, when you get to the muscle fiber level, that's where the cells, that's a cell, but it's really hard to see where the cell starts and where it ends because they're really long and they're just made of all these little myofibrils. Okay, so now we're going to look at what we call the sliding filament model, and that's how muscles contract. And this is how skeletal muscles contract, but the other ones, there's similarities and differences. We usually just look at skeletal muscle. So skeletal muscles are long cylinders, multiple nuclei. It's hard to know what one cell is. You have a sarcolemma, which is right beneath the endomysium, and it's like another, it's like the plasma membrane. It's like another layer. And then the sarcoplasm is the cytoplasm. There's glycosomes, which contain glycogen, which is glucose. It's a form of glucose for energy. And then you also have myoglobin. Remember, myo, that prefix means muscle, and that stores energy. So you have like oxygen. So you have oxygen and glucose readily available on site for the muscles to use to make ATP and contract. And then you have some modified organelles. There are myofibrils, there's sarcoplasmic reticulum, which is kind of like the endoplasmic reticulum, but sarco means muscle. And then there's T-tubules, and those are part of the cytoskeleton. So myofibrils are rod-like elements. They are made of protein, and they have striations, sarcomeres, myofilaments, and molecular composition of the myofilaments. So this is one myofibril right here. Muscle cells also have a ton of mitochondria so that they can metabolize that glucose with the oxygen and make ATP. Here is your sarcolemma. That's like your cell membrane. And then on the outside would be your endomysium. This is the part that makes these muscles striated, is the myofibril. They have these little stripes on them, and that's actually where the contraction is happening. So let's dig into that. You have these little striations. These are repeating dark and light bands along the myofibril. And the A bands, you'll see different names for these in different textbooks, but in yours, the A bands are the dark regions. And then the H zone is a lighter region in the middle of the A band. The M line is like an anchor. It's a line on the protein that is right in the middle of the H zone. And then the I bands are the lighter regions, and they have a Z line. So there's the M line and the Z line. These are like zigzag little pleats on the light part of the myofibril. Okay, let's see what it looks like. So this is one muscle fiber. This would be part of a cell here, weird looking cell. And there's some nuclei, and then these are the bands. You can see the dark A band and the light I band. This is what makes it look stripy and striated. And this is what's actually going to do the contracting. So when your skeletal muscle contracts, you have millions of these little dark bands and light bands, and they're going to move closer to each other when the muscle contracts and then further apart when the muscle relaxes. So I'll show you what that looks like in a second. One sarcomere is one piece, like one of those little stripes. That's the smallest unit. It's kind of like, we haven't learned about alveoli and lungs yet, but it's the functional unit of the muscle. It's the one little worker that's doing the work to contract the muscle, and they all work together like synchronized little swimmers, and they all contract at the same time, and that's what makes the muscle contract. So it has an A band and half of an I band, and it's just like one little piece. It's like boxcars of a train, and they're going to all squeeze together at the same time. So this is a zoom in of that. This is one H zone and another H zone. Right here is your M line, like these triple things here are your M line, and then this is your Z line. Okay, so one sarcomere is from here to here, from like the Z to the Z. Z line to Z line, that's one sarcomere. So on a myofibril, you'll have a whole bunch of Z lines. Oops, I just clicked. You'll have a whole bunch of it end to end, and then when the muscle contracts, all of those are going to squeeze the whole length of the muscle fiber, which many fibers make up a cell, the muscle cell, and they're going to squeeze together and contract. So you have these different parts. We're going to zoom in even further on how this works. You have these myofilaments. They are made of two different kinds of filaments, actin and myosin, that are in a sarcomere. And this is what makes up the A line and the A band and the I band and the Z line and the M line. There are these little filaments. So the actin filaments are called thin filaments. That's a synonym. They are anchored to the Z discs. So the actin filaments anchor to the Z discs. And then the myosin is the thick filament, and they are connected to the M line. Okay. So these two are the ones that are interacting. You've got the myosin and the actin are the ones doing the moving. They're the ones acting, and the myosin is going to be the like the anchor. Okay. So here is your M line. This is your myosin. Let me get my laser pointer. This is your myosin. That's the thick filament, and then your Z line is your thin filament. This blue right here. Okay. And then your myosin is anchored to the M line, your actin is anchored to the Z line. What happens is these little heads, these are called myosin heads right here, these are going to lift up and grab onto this actin filament and pull it in. So the actin is going to be pulled in and move. The myosin is staying put and it's just grabbing that. So the red little knobs are grabbing the blue actin and just pulling it in kind of hand over hand. And that's going to shorten that distance of that Z disc. That is one sarcomere from Z disc to Z disc, or Z line to Z line. That's going to shorten, and that's causing the muscle to contract. And when all of them do that along the length of a muscle fiber, your whole muscle is contracted. That's what you're doing right now when you're moving, like I'm moving my arms. All these little Z lines are moving closer together because they're being pulled by the myofilament, the myosin, and then that whole Z line gets shorter. The whole sarcomere gets shorter, and millions of them all get shorter, and that causes your muscle to contract. Okay, so thick filaments, are composed of myosin, and they have these little heads, and they form cross bridges with the actin. So the cross bridge is where the little myosin head is grabbing the actin, and it's going to pull it in. And that action is called a power stroke. And then when that myosin head goes back down, that requires ATP. So then you have your actin These, and all of this is made of protein. This is why if you want to build muscle, you need protein, because that's what your muscles are made out of. Okay, so there's more to it than this, actually, like on how the myosin actually grabs the actin to pull it in. You have these little proteins called tropomyosin and troponin, and they are covering up the little spots on the actin where the myosin can grab. So let me see if there's a picture here. So here is your little myosin. This is your this is your thick binding site. This is the myosin. These are going to grab onto these binding sites here. Or this guy is this. Yes, I don't know, it looks upside down to me. But this little guy is this. This is going to grab onto an actin binding site, the little blue things, right? But there are proteins that are in the way on the actin. And the only way they get out of the way is if calcium shows up. So here is the actin, the blue. The little myosin heads want to grab onto, where are they showing the binding sites? Like probably these little circles, yeah, these little empty circles. But right now they're blocked. So this is where the myosin wants to grab and pull the actin in to contract the muscle. But right now they're blocked by tropomyosin and troponin. So what has to happen is calcium's going to come in, it's going to bind to the troponin, then the tropomyosin will move out of the way, and then the little myosin head can come in and grab on these little nuggets right here and pull the actin in. So that's why we need calcium. That's one of the reasons why we need calcium. Our muscles cannot contract unless calcium binds to the troponin, moves the tropomyosin out of the way, and then the muscle can be pulled. Okay, and there's like really good videos on YouTube that really show this. Sliding filament theory is what we're looking at, so I would recommend watching those. And I think it's very cool. It's cool stuff. Okay, there's also other proteins that do the work too. There's an elastic filament made of titin. It holds the thick filaments in place and helps them go back to normal. And then dystrophin links the thin filaments to the sarcolemma. Then you have nebulin, myomycin, and C proteins that bind the filaments and sarcomeres together. So there's a lot of players here that are working together. There are, of course, disorders that, like with anything, that if something is wrong with this mechanism, you're going to have a disorder. So one of them is Duchenne muscular dystrophy, and it's a really bad form of muscular dystrophy where your muscles are destroyed, and it's a sex-linked recessive disorder. Mostly males, because it's sex-linked. Males only have one X chromosome, so if you have that allele, you have it. Appears between 2 and 7. Kids are falling often. If you have good care, you can live past 30s. And then it is caused by a defective gene, dystrophin. So what that means is the protein that links the thin filaments to the extracellular matrix and the sarcolemma is dysfunctional. So your muscles are not, like your thin filaments are not anchored correctly. It allows you to have excess calcium. Your muscles tear easily. So like the muscle contraction mechanism doesn't work right. So here's a child with that disorder. Okay. Oh, this is weird. I think we already kind of have that information. Okay. Now we're going to look at like more on the muscle contraction, because you don't actually, like, we haven't covered everything on how the muscle contracts. So remember the sarcoplasmic reticulum, which is a modified endoplasmic reticulum, just in muscle cells. It is this unique structure in muscles where it kind of surrounds the muscle fibers, and it stores calcium in it, and it causes the calcium to be released. So what happens is when the nerve sends an action potential saying like, hey, this muscle needs to contract, that causes the sarcoplasmic reticulum to release calcium, and then the calcium floods the muscle fiber. That binds to the troponin. Tropomyosin moves out of the way. The myosin head can come in and pull the actin and contract the muscle. You also have T tubules. Those are like part of the sarcolemma. They make this triad. So like every muscle fiber is just wrapped in the sarcoplasmic reticulum, just all the way around. And then that's what's going to cause when it receives an electrical impulse, the calcium floods out, and then it causes the muscle to contract. Okay, so that is part of the sarcoplasmic reticulum. The T tubule is what regulates that. So that's interacting with the action potential in the nerve. Okay, so it receives the electrical impulse. T tubule changes shape. It allows the sarcoplasmic reticulum to change shape and releases calcium. So here is the sarcoplasmic reticulum that's like covering every muscle fiber. It is modified endoplasmic reticulum, and it's like, look at it, it's all the way around like a net. It forms the T tubule is like this lighter blue part here, and so this is the triad, like 1, 2, 3 parts here, boom, boom, boom. The T tubule is what gets zapped, so we're not really showing that. It's not showing the nerve, but it's going to get zapped, and then that's going to cause calcium to flood out of the sarcoplasmic reticulum. That's going to go into these sarcomeres, into these muscle fibers. So calcium's going to flood, and that's going to cause the muscle to contract. So contraction is when that cross bridge from the myosin grabs the actin and pulls it in hand over hand. Okay, that's a contraction. And all of those sarcomeres along the length of your muscle fiber are going to contract. That is the siding filament theory. And that part is where the calcium binds to the troponin. Tropomyosin moves out of the way. The myosin head grabs the actin, pulls it in. That's all the sliding filament theory, and it forms cross bridges and pulls the actin in. Okay, so I think I've explained this like 14 times. It's hard to know what order to explain this mechanism in, because like this has to happen and then this has to happen, and you kind of keep going backwards and backwards, but it's just a lot of information to pile on. So here is Z line to Z line. This is one sarcomere, right? So here's a real picture of it, and then this is the M line right in the middle. So this is when it's relaxed, okay? And when the nerve impulse, like when you receive an action potential, this isn't shown, but you have sarcoplasmic reticulum outside of this, like way outside. It's wrapping around all of these muscle fibers. When it receives an action potential in the T tubule, then that's going to pass to the sarcoplasmic reticulum. That's going to release calcium into this space. The calcium is going to bind to the little troponins, which they're not showing here. The tropomyosin is going to get out of the way, and then this little myosin head can attach to the binding sites and pull this in. So then, I'm trying to show you with my hands, the whole like Z line to Z line is going to shorten and get closer together, closer to the M line. So the sarcomere length shortens. Oh, I think the slide after that would have just shown the shortened version. So check that out, because like you won't get it unless you see the shortened version. Okay, so there's more to this, but wait, there's more. So the neurons are working closely together with the muscles to make the muscles contract, and this happens at what we call a synapse. And I know we haven't done nervous system, so it's hard to know some of this, but AP is an action potential, and that's the nerve impulse, and it's going to go from the neuron to the muscle, but there's a gap in between the neuron and the muscle, and that's called the synapse, and it's going to release a chemical in that gap called acetylcholine. There's also ion channels that are involved. We'll learn about this also in the nervous system, but along the membrane of the nerve, there are these little ion channels in the membrane that let stuff in. They let ions in because otherwise the ions can't get in. You have chemically gated ion channels and voltage gated. Chemically gated means if there's a chemical that comes in and binds to a receptor, then that gate's going to open. It's like the key. Voltage gated is it's going to open when the charge in the cell is a certain amount. That's going to cause the ion channels to open. Okay. So this is a chemically gated one. So here's your cell membrane. These ions are trying to get in or out, but they can't because they can't get through these phospholipids. And so you're going to have this chemical messenger that's going to bind to this gate and cause it to open and let the stuff in or out. A voltage gated channel, when this is a certain voltage inside the cell, this little plug is going to come off. This channel is going to change shape, and then stuff is going to come in. Okay, so those are like two players that help this whole nerve impulse thing happen. Okay, so nerves are made of cells too, and they are kind of a long, skinny cell, and they have these axons, which is the long, skinny part of the cell. So they have a cell body with like a head and then a tail, and the axon is the tail part. The tail ends when we're dealing with nerves that talk to muscles. Because some nerves talk to nerves or talk to glands. So when you're doing the motor neuron that talks to muscles, the axon ends at a muscle fiber, and that's called a neuromuscular junction or a motor end plate. So this is where the nerve interfaces with a muscle. So here it is. Here is a nerve, and then it is touching a muscle. These are the nerves that are going to tell these muscles to contract. Okay? So this spot right here is a neuromuscular junction. Okay, you have an axon terminal, which is the end of the axon, and it ends at this synaptic cleft, which is this space, like a gap where it goes from the nerve to the muscle. That is going to tie into the sarcoplasmic or sarcolemma and eventually into the sarcoplasmic reticulum. And you're going to have a neurotransmitter, which is that chemical called acetylcholine. That's what's going to tell the cell, like, hey, this muscle needs to contract. Okay, so this is the end of the neuron here, and then this is the sarcoplasmic or sarcolemma of the muscle. There is a space here, so these are not actually connected, they're just like near each other. And then these little green things are the neurotransmitters. These are the acetylcholine. They get released, like when there's an action potential coming through this neuron, then it releases this acetylcholine. It's going to bind to these little receptors, and it's going to open up those gates. So when the nerve is carrying a signal and it gets to the end, it's going to release these little chemicals. They're going to stick to this muscle, the sarcolemma, cause the gates to open, and then that's going to start the sequence of events that will cause the calcium to flood the muscle and cause the muscle to contract. Okay, so big picture. You have the neuromuscular junction releasing acetylcholine. The muscle fiber becomes excited. So it actually continues a nerve impulse. Like that nerve impulse goes through the muscle fiber too. You have this excitation, contraction coupling, and then the calcium is released, and then that causes the myosin head to bind to the actin and pull it in. Okay, so here is your neuromuscular junction right here. What's happening here is the action potential is moving down the axon. It releases acetylcholine. It binds to the little receptors, and then that causes this end plate to get an action potential through it. Like this gets a nerve impulse, this muscle. It causes the inside of this to become depolarized. That action potential goes down the T tubules. Then that zaps the sarcoplasmic reticulum and causes it to release calcium. The calcium binds to the troponin, moves the tropomyosin, uncovers the myosin binding sites, myosin grabs actin, and the contraction happens. So those are all the steps. So we kind of started here, and then I was like, but wait, there's more. There's this calcium situation. But wait, there's more. There's this sarcolemma. But wait, there's more. There's this nerve. So we kind of went backwards, but this is actually the order that it happens in. So here's another list of those things. So you have your action potential arrives, voltage gated channels open, and and calcium enters the motor neuron. I kind of skipped that part, but that's what causes the acetylcholine to leave and then bind to the receptors on the muscle side. Then that causes gates to open, sodium rushes in. This part I also skipped. This is interesting. So the green stuff was the acetylcholine. It's in the synapse. It's the stuff that binds to the receptor. You have an enzyme that's going to break down that acetylcholine after it's bound. Because if it sits around in that synapse, the muscle's going to keep contracting. You're going to get a muscle spasm. So you need this acetylcholinesterase to destroy the acetylcholine so that your muscle only contracts for that moment and it doesn't keep contracting and cause a muscle spasm. Okay, so here is that again. So here's your motor end plate, your neuromuscular junction. Calcium actually comes in when that action potential is traveling down. That's what causes acetylcholine to come out. Acetylcholine binds here. That causes sodium to come in, and that's going to cause the T-tubule to be zapped, which will cause the sarcoplasmic reticulum to release calcium, which will cause the muscle contraction. But here is the acetylcholine sitting right here. If it stayed there, you would have a problem. Your muscle would keep contracting. So that's why we need the acetylcholine esterase right here to break down the acetylcholine and get rid of it. So your muscle contracts and then it's done. You don't keep contracting or you're going to have problems. There are diseases where, like, I think it's maybe a malfunctioning acetylcholine esterase, or for some reason your acetylcholine doesn't break down. I think it's called myasthenia gravis. And you keep getting spasms because the acetylcholine keeps causing your muscles to contract. Okay, so watch this. This is probably a really helpful video because I know you're probably really overwhelmed and confused. Okay, there are drugs, toxins, diseases. Myasthenia gravis, there it is. It's because you have a shortage of acetylcholine receptors. This is an autoimmune disease, so your receptors are not working correctly. It's going to cause drooping eyelids, difficulty. Oh, it's muscle weakness, so it's the opposite. It's your muscles aren't contracting enough. And if you take like a muscle relaxer or things like that, they're probably acting on something with that mechanism. That's probably how they're doing their job. Okay, when the sarcolemma is resting, it's polarized. So when it's not contracting and there's no like contraction, no action potential, it's polarized. And then it's going to become depolarized when it's being zapped. So there's 3 steps to that. And when it's being zapped, it is electrical. Like just like your nerve impulse is electrical, when the muscle is being zapped, it's an electrical response too. So 3 steps: generation at end of plate potential, depolarization, and then repolarization, and it goes back to normal. So those are the same steps basically as a nerve that's having an action potential. Okay, so once again, action potential comes down the nerve, calcium rushes in, acetylcholine is released, binds to the receptors. That causes sodium to go in. So here's your little wave of depolarization. That's going to cause sodium to go in. This little channel is closed so it doesn't let the potassium leave until depolarization is done. And then once it reaches a specific charge, like voltage, then the potassium is going to get to leave. So these are voltage gated channels here, and they're only going to open or close if the voltage is a certain thing. Okay, so end plate potential, acetylcholine is released from the motor neuron. We said that 14,000 times. That causes a chemically gated ion channel to open. So that's the ligand, that's acetylcholine binding. That's what's causing it to open. Then sodium diffuses into the muscle fiber. Some potassium might leak out, but not very much. Since sodium diffuses in, it becomes more positive inside, and that is the depolarization because it's more positive inside. So depolarization, once it reaches a certain point, the voltage-gated sodium channels will open. You'll get a whole bunch of sodium inside, and it becomes fully depolarized inside, meaning it becomes positively charged inside. Then once it reaches a certain positive charge, the sodium gates close, the potassium gates open. And this is because it's good, like it did the thing, and now it's going to reverse it and reset that the sarcolemma. So then the potassium voltage gated channels will open and potassium will leave. And it also is going to overshoot. So it's going to become negative inside because potassium is leaving, and it's going to overshoot and then go back to normal. Let me show you with a graph because you don't know what I mean by overshoot. So it's not showing it very well, but so it starts out negative inside this muscle, also inside, like muscle fiber, also inside the nerve. This is exactly how nerves work too. They start out negative, so you can see negative 90. When the action potential starts, that's going to cause the sodium channels to open, and then the charge is going to go up because they're flooding with sodium, okay, inside the cell. Once it reaches a certain point, the sodium channels are going to close and the potassium channels are going to open, and then potassium is going to leave the cell, and that'll drop the charge, and sometimes it overshoots a little bit and then comes back to normal. Okay, so that's what's happening in the muscle fiber when it is contracting. This action right here is causing it to zap the T-tubule, which causes the sarcoplasmic reticulum to open up and release calcium on the muscle fiber. Whew, I know it's a lot, but it's cool. So excitation contraction coupling is when the action potential is causing that sarcolemma thing that I just said. So it's causing the calcium to release. Okay. So here's your motor end plate, acetylcholine. It goes in, it causes all that like positive, negative business that we just saw, zapping the T-tubule, so making it positive in there. That zaps the sarcoplasmic reticulum, causing calcium to flood out. And this is why we need calcium, potassium, and sodium. Okay, here's a zoom in of that. And like I said, there are tremendous videos that show this, so watch them because this is complicated. So you have your action potentials running through this T-tubule, and then that actually causes these little gates to open, and it releases calcium into the muscle fiber, and then finally, we have a picture showing the calcium binding to the troponin. See how the troponin was right here and the tropomyocin's blocking those little binding sites? Now calcium bound, it moved the tropomyosin out of the way. Now your myosin head can bind. End of the story. That's the whole picture. Holy moly. Okay, watch this video again and again and again until you get this. Okay, so the cross bridge doesn't happen very well at low calcium because the tropomyosin is blocking and all of that. When you have high calcium, then it's going to be able to bind and pull the actin. And if you don't have enough calcium, your body's going to steal it from your bones. So make sure you have enough calcium. When the action potential is gone and you no longer need to contract your muscle, the calcium goes back into the SR, the sarcoplasmic reticulum, and waits for the next one. Okay, cross-bridge formation, myosin head attaches to the actin filament, and then you have the power stroke where it pulls it and shortens it, and then with the point at which ATP is used is right here. So ATP attaches to the myosin head and causes the cross-bridge to detach. So the cross-bridge attaching does not require ATP. It's the detachment that requires ATP. So, okay, and it gets it ready for the next one. So here that is, we have calcium, so it gets the tropomyosin out of the way. It's going to bind to the binding site, and then it's pulling the actin in, contracting the muscle. And then when, what's that showing? When it needs to detach, that's when you're using ATP. If you don't have ATP, then the head will not detach and your muscle will stay contracted, and that is what rigor mortis is. So like in someone who has died and has stiff muscles, it's because that myosin head did not detach. Okay, so watch that. Okay, so rigor mortis happens 3 to 4 hours after death. Your muscles stiffen. It's the worst at 12 hours, and then they relax again. And it's because you have a ton of calcium levels, but the ATP isn't being made by your cells because your cells are dead. And so your muscles the myosin head is not able to detach. So you have cross bridge formation, but not cross bridge release. And that's it.